In parts 1 and 2 of this lecture on chapter 2, we have looked at how elements from the periodic table are able to interact with other elements to form molecules, so how those atomic interactions happen and how molecules are formed. And in this last section, we're going to see some examples of some of the major molecules that make up living things, the ones that we're going to see repeatedly when we look at what cells are made out of and how things move around the human body. And the majority of these are what we call macromolecules. And that is exactly just as the name implied, macro meaning large, and molecule meaning molecule, so large molecules. And most macromolecules are so large because they are what we call polymers, which means they are composed of many repeating subunits that form a long chain of molecules that become bonded together into a larger molecule. And each one of those repeating subunits of a polymer is a monomer. Mono meaning one, so one subunit of that large chain. Poly is many, many repeating subunits. So if you look at our Lego example here, each of the individual Legos that make up the chain are monomers, the individual repeating subunits. And then when you add all of those monomers together, then you get a growing chain of a polymer. And many of the substances that we store within our body, like sugars and fats and things like that, are going to be stored as chains of monomers into these larger polymers. So you could think of the individual Legos as being a sugar like glucose. And then as you grow a larger chain of sugars, you're going to get larger, more complex sugars. And that happens to be our first major example of a molecule of life. But let's look at how those repeating subunits are either added together or broken apart first. So the monomers that make up a polymer, when we want to add them together, join them and bond them together, we do that through a reaction called dehydration synthesis. Dehydration synthesis. So this top reaction is adding two individual monomers that are right now are separate from one another, but we're going to create a bond right here to bond them together and start to create our chain that can grow into a polymer. The dehydration synthesis reaction is also sometimes called the condensation reaction, but whatever you decide to call it, the important thing to remember is that you're losing water. So you're going to have water as one of the products at the outcome of your reaction. And then this example, we have two molecules of glucose and their hydroxyl group, this OH group that's hanging off of the end here. So two hydrogen and one oxygen are lost, and that is what creates that extra molecule of water over there. The oxygen's octet is still complete by forming that covalent bond with those two carbons, one from each of those original glucose molecules. So in the end result is you have this molecule of water, dehydration or condensation, and you've joined your two monomers together into a chain. If you wanted to reverse that process and actually split those two monomers apart and end up with two individual pieces, that would be hydrolysis. And that lysis, that ending there, lysis means splitting. So this is literally water splitting. Hydro, water, lysis, splitting. So we're going to split this molecule of water to complete and go back to our hydroxyl groups on our individual glucose molecules. Everyone's octet is still met, but we're just reconfiguring how those electrons are shared. So the dehydration synthesis reaction will add monomers together to create that chain, and the hydrolysis reaction will split them apart to again go back to our individual individual monomers, or the pieces that are the repeating subunits. So our first example of our major molecule of life are our carbohydrates. And carbohydrates are sugars and starches that are primarily served to make energy for our body. And they're made entirely of carbon, hydrogen, and oxygen. And they are generally classified by how big they are. So what's their overall size? Monosaccharides. Saccharide means sugar, and a sugar is a type of carbohydrate. So monosaccharides are simple sugars, and these are our smallest molecular units that are carbohydrates. And they are the monomers that join together to form those long polymer chains. So glucose, our example glucose on the last slide, is an example of a monosaccharide. Oligosaccharides are polymers made from chains of monosaccharides. Oligo means few. So when two of our glucose molecules were joined together through that dehydration synthesis or condensation reaction, they formed a molecule of maltose. And this is an example of an oligosaccharide. Lactose is another example that we'll see this semester, made of one molecule of glucose and another molecule of another sugar called galactose. So carbohydrates, they don't have to be formed from chains of the same monomers, but they're going to be classified by how big they are overall. And then our last kind of form of carbohydrate is a polysaccharide, and these are our complex carbohydrates. And these are made of very long chains of monosaccharides, and they get so large that they start to branch off of each other. So if this was a complete picture of a polysaccharide, you'd see branching sugars going off in all directions. And that's glycogen. This is how we are storing a lot of our energy reserves in this form of complex carbohydrates. And then as we need them, we can use that hydrolysis reaction to split off these individual monomers and use them for energy. Lipids is our next group of major molecules. We did talk in the last video about a lot of the properties of water and about it being a universal solvent, but of course there are always exceptions to every rule, and lipids are a really, really important example of something that will not dissolve in water. Lipids we know that, right? Oil and water do not mix. Lipids come in a couple of different types: triglycerides, steroids, and then the all-important phospholipid that we'll talk about next. Phospholipids are I would say the most important group of lipids, especially that we're going to talk about this semester. And they are composed of a single molecule of glycerol. So that's going to be like our glycerol head here, this ball piece. And then they have two fatty acid tails. So unlike our triglycerides that have 3 fatty acid chains, phospholipids are going to have 2 fatty acid tails. And they have a negatively charged phosphate group on that glycerol head. The charged phosphate group gives it that polarity, and that fatty acid tails are nonpolar, and that makes them what we call hydrophobic or water-fearing. So they're going to not be attracted to water. They're going to try to avoid water. The glycerol head of the phospholipid is polar, and that section is going to be attracted to those polar bonds that we saw in the water, and those are going to orient themselves towards water. It's these forces, having the hydrophilic end and the hydrophobic end, loving water or wanting to avoid water, it's those forces that enable our cells to maintain their structure and keep certain substances in and keep certain substances out. Because surrounding each one of our cells, we're going to see what we call a phospholipid bilayer, 2 layers of phospholipids. So basically, all of our cells are surrounded in this type of fat, and it's the interactions with water that allows us to keep and maintain that shape. And then that phospholipid bilayer is going to create a substrate or an area for all of the substances that need to move into a cell or out of a cell to go through. We're going to come back and talk about that in a lot more detail. So definitely lock away, what is a phospholipid and how does it interact with water? Because we're going to see that again next week when we talk about the structure of our cells. Our next group of molecules that we'll see repeatedly are our proteins. And proteins are polymers or chains of amino acids. And these can create one or more chains. Amino acids are the building blocks of proteins that contain a single carbon bound to a hydrogen with an amino group, and then the carboxyl group, plus a group that will vary from amino acid to amino acid. You do not have to, for this class, you do not have to memorize amino acids. If you go on to take anatomy and physiology, that will change, but for this semester, you don't need to worry about that. Some amino acids are what we call non-essential, and some of them are essential. So non-essential does not mean that we don't need them to survive, but non-essential means that we can make them on our own. The ones that are labeled essential means that our bodies are not capable of synthesizing them, so we have to obtain them from food that we eat. So if it's an essential amino acid, that means that we have to consume that in order to have that product to build our proteins. If we don't eat it, then we're going to be deficient in that amino acid, and we're not going to be healthy, and we're not going to be able to build the proteins that we need within our body. The structure of a protein is really important to ensure that it functions correctly within the body. So the shape and orientation of the different, what we call reactive groups, influence how other molecules will be able to interact with that protein, and will it be able to do its job within the body, essentially. So the primary structure is the specific sequence of amino acids that make up the chain, and that is determined by our DNA. That's the information contained within our DNA. And then the secondary structure are the shapes that the chain forms based on the hydrogen bonds that are formed by amino acids next to each other. And that's those hydrogen bonds, because DNA itself is a protein, those hydrogen bonds are what gives DNA its helical shape. So a helix is one of the shapes that a protein can make. It can also make what we call a pleated sheet. But those shapes are influenced by the hydrogen bonds. The tertiary structure is the 3D shape that the protein forms. So when proteins are moving throughout our body, they kind of look like blobs. They kind of fold up on each other. And those are, again, formed by additional hydrogen bonds. And if we see a phenomenon called denaturation, that means that the protein has lost its 3D shape, lost its tertiary structure. And if that happens, then that protein will no longer function within the body. It won't be able to do its job. And that won't be able to do its job because when a protein is folded up properly in its tertiary structure, the portions on the outside are the portions that are doing the work within the body. And if those aren't accessed or accessible by enzymes or the other molecules that it needs to interact with, if they can't get to it, then that protein won't be able to do its job. Heat is the most common thing that can cause denaturation or the loss of that tertiary structure. Not all proteins are going to have what we call quaternary structure, and that only happens if there are more than one chain that are housed within the protein. Our last kind of group of molecules are enzymes, and enzymes are most often proteins, so I'm going to lump them in with the proteins. And we are going to talk more about enzymes when we get to an example of their function, but I kind of wanted you for now to be aware that they exist and that they are most often proteins. So we're going to include them in that group.